Hello everyone, this is your instructor Kwame Shen, and this is part 2 of our lecture on microbial genetics. For part 2, we will be looking at transcription, that is from DNA to messenger RNA, and translation from RNA to proteins. So it brings us back to the central dogma, how we move from genes, from DNA to proteins. We'll look at transcription first. Because transcription involves the copying of DNA into complementary copies of RNA, we need to appreciate the difference between the two. For DNA, the sugar is deoxyribonucleic acid. For RNA, it is ribonucleic acid. And it comes down to the second carbon in the sugar structure, that for deoxyribonucleic acid, it has only hydrogen attached to the second carbon. For ribonucleic acid, it has OH attached to that. And then for the structure, DNA is double-stranded generally, so it has two strands. RNA has only one strand. There is also a difference in the base pairs in terms of the nucleotides that join or are attached to the strands. Note that for all organisms, this backbone, either DNA or RNA, is the same. It's a phosphodiester bond that is formed between the phosphates and the sugar. However, the nitrogenous bases will continue to change, and it's the changes that results in different proteins, because the changes results in different genes, so different proteins, and therefore different instructions for the cells. For RNA, the base pairs are the same, except that uracil replaces thymine. But the rest are all the same. So the same point that I made previously is what I'm making. The difference between deoxyribo and ribo is that for the ribonucleic acid, there is OH at the second carbon here. But the rest are the same. You have uracil replacing thymine, but it's single stranded, but the other information is all the same. There are 3 types of RNA, and the types are based on the different roles that they play in protein synthesis. So there is rRNA, or ribosomal RNA, that plays a part in the structure of the ribosome itself, which is the site of protein synthesis. And then you have tRNA, which is transfer RNA, and what it does is that it brings the right amino acids to the ribosomes and then binds to the ribosomal surface and ends up helping in the formation of the polypeptide chain or the protein. Then you have the messenger RNA, which is the code. It contains the information from the DNA, which would direct the synthesis of proteins. So you have these 3 different RNAs. So the process of making an RNA, which would have complementary base pairs. So this word is essential. They are complementary, and they are formed from a template of the DNA. So you have copies that match up. So if you have adenine, for RNA you have a uracil attaching to it or combining with it, and so it's going to be complementary that way. I'll show you what I mean in a moment. But for that to happen, you will need an enzyme, and the enzyme that you need is the RNA polymerase. Remember, we've already seen DNA polymerase. Now it's RNA polymerase, and RNA polymerase will help synthesize a new strand of messenger RNA that would have base pairs that are complementary to the DNA. Note that DNA cannot be read directly into protein, so it's essential that we have an intermediate, which is the messenger RNA. So you would have a DNA strand, double stranded like that, and then the area where the transcription would take place, there is an indication, and polymerase will attach, RNA polymerase will attach to that area. It will cause that area to unwind, so there is opening up of that section, and then the synthesis will begin. So this is the template strand. This strand is the template strand. That is the strand that the complementary bases are made from. What I mean by complementary bases is that if you have C or cytosine in DNA, you would have G being formed on the RNA. It's complementary to this. If you have A, you have U. Because it's RNA, you have U, not T. So that's complementary to it. Like that, if you have T, you have A, complementary to. You end up forming the complementary base. It's being formed in this direction. And so the chain is formed behind it as it moves forward. So this is the template strand. This is the non-template strand. There is more information you can get out of this diagram, but I'll talk about that in a moment. But the process takes 3 steps. There is initiation, there is elongation, and then there is termination. So initiation is when you have the RNA polymerase binding to the specific site. The RNA polymerase is guided to bind to that site by a molecule called the promoter. Okay, so the promoter will guide the RNA polymerase to bind to a specific site of the DNA. And then once it binds there is unwinding, and then RNA nucleotides are synthesized. That's that process known as elongation. And then it gets to a point where there is a stop codon, there's a stop information, and at that point, the synthesis is brought to an end. So you have the process happening in this way. So you have initiation, where you have the RNA polymerase binding to a promoter region. So there's a promoter region causing the unwinding. Then nucleotides are added by the polymerase. So there's a direction of synthesis. It's unwinding will continue to take place. New nucleotides are added. Messenger RNA is then being synthesized until it gets to a point where it's indicated that it has to terminate it. And then you end up with your RNA strand. So transcription is generally moving from DNA to RNA. And so you have a DNA double-stranded. This is the template strand. So the messenger RNA you have here is complementary to this. So G with C, T with A, like that. It's the same process that is being summarized over here as well. You end up having your transfer RNA at this stage. So I need you to practice this. If you're given a messenger RNA strand and you are to make a copy, if you're given a DNA strand, there's a DNA strand, and to make a copy of a messenger RNA, you should be able to do that because remember the strand is complementary. What you need to have a remember is that in places where you are to form a complementary with adenine, you cannot have T. You need to have U in place of T. So the U will be here. You would have C. You would have another C here. You would have G. You would have A here. And then you have A here. So continue like that and do same for this. Then there's translation from messenger RNA to protein. From messenger RNA to protein is known as translation. So that is translation. So you have your DNA, it's been transcribed into messenger RNA, and then from messenger RNA to protein or polypeptide, and that's translation. So how do we go from RNA to amino acids? Now that you have the RNA, the cell should be able to read that information and be able to interpret it. So it's like you're translating a language, be able to interpret it and then instruct the cell to form a particular sequence of amino acids. So that's what translation is about. And so that leads us to what we call the genetic code. The codons are 3 nucleotides, so groups of 3 nucleotides. So what the cell does is that it reads the information on the messenger RNA in threes. So 3 nucleotides are read together. 3 nucleotides will code for 1 amino acid. 3 nucleotides, 1 amino acid. And these 3 nucleotides are called codons. So the 3 nucleotides on the messenger RNA are called codons. And if every 3 is coding for a particular amino acid, then we can actually come up with a code. We know what each of them will code for. And that is what we call the genetic code. And so you have the 4 nucleotides, uracil, cytosine, adenine, and guanine, are all combining. And they can combine, you have 64 possible codons, or 3 readings of 3 combinations. 61 of them will code for amino acids, including a start codon, and 3 of them will code for nonsense codons. We call them nonsense codons because once they are coded, it results in a stop codon. Okay. So there is something going on here. We only have 20 amino acids. Why do we need 64 or more specifically 61 different combinations of amino acids that can be synthesized from this? Okay, and this results in what we call redundancy, and this is what I mean by that. So this is one amino acid. It's called phenylalanine. You can see that you can have 2 different codons coding for phenylalanine. You have UUU and UUC that will code for phenylalanine. Why don't we just have UUU and that's it? You have phenylalanine. You don't need 2. And then you have leucine. You have UUA, UUG, leucine again, CUU, CU. So you have 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. 6 different codons coding for leucine. Why is that even important? So this is what we call redundancy. Okay, we call it redundancy. So you have redundancy built into the genetic code. These are the stop codons. So UUA, UAG, and UGA. And this is the start codon, AUG, which codes for methionine. So you have messenger RNA being translated into protein. If you have AUG, that's a start codon. You have 3 of them. So you should know where the synthesis is going to begin. It's going to begin with a start codon. You have CCG, that's proline. You have CAG, so on and on and on. So they are translated into threes by threes. So read by threes. Those are the codons. And so where the transfer RNA comes in is that, so you have these codons on the messenger RNA. You have these codons on the messenger RNA, as you can see down here. The transfer RNA is the molecule that's going to bring the amino acid. So on its surface, it also has sequences of nucleotides that has to be complementary to the messenger RNA. And those sequence of nucleotides are called anticodons. So there are 3 groups of nucleotides that are complementary to the messenger RNA. And they on the transfer RNA, they are called anticodons. So that's an anticodon. And this anticodon, so you have a codon here. 3 nucleotides on the messenger RNA. You have anticodon, 3 nucleotides on the transfer RNA. And they're going to bind, so you have G with C, A with U, G with C. It's complementary. Okay. this is where the amino acid will be attached to. So immediately that it recognizes this particular sequence on the messenger RNA, it's complementary to it to attach to it, and therefore it brings to it a specific amino acid. So this is how, pictorially, this is how the process happens. You have the ribosomal subunits, you have the small units and the large units. The messenger RNA, which contains the start codon, messenger RNA contains the start codon, will be here. You would have transfer RNA that has complementary codons. So you have G, that's C, you have A, that is U, you have U, that's A, complementary, and it will bring a particular amino acid. In this case, that's the start codon, so it's going to bring methionine. And so it's going to bring it and attach to the ribosome. The spots that it attached to, the site that it attached to, is known as the P site. So it touches there, it has its amino acid attached, and there's a complementary, it's complementary to the start codon on the messenger RNA. You would have a second transfer RNA also come in, bringing another amino acid, and the anticodons would also be complementary to the codons, the second codon and the messenger RNA, so that it can attach over here. Okay. Now, the site that the second transfer RNA will attach to is known as the A site. So you have the P site, you have the A site. Once it attaches to its complementary codon nucleotides, it will then form a peptide bond between the two amino acids. So the two amino acids between the first transfer RNA and the second that just joined will form a bond known as a peptide bond. Meanwhile, the ribosome is moving along the length of the messenger RNA. So it's moving in this direction. So it will continue to move. Once it moves, it's going to push the first transfer RNA that has the start codon, that's attached the start codon, to the next site on the ribosome. And that site is known as the E site. So this particular amino acid leucine that's attached to the second transfer RNA will then be moved to where you originally had the P site, and then there will be a space created for this amino acid, the next amino acid, to be added. So you have the third transfer RNA attaching to its complementary codons, and then it will bring also with it its amino acid, and then it will also form a peptide bond like that, another peptide bond. And so then it's still moving along, and so the process goes on and on and on until you have a stop codon. And once you have a stop codon, what will happen is that the synthesis will cease, the last transfer RNA will also be released, and then the ribosome will separate. Then you end up with your new protein, like that. In prokaryotes, this is how the process happens. So there's a direction of synthesis. You have your DNA strand, that's the green one, of a more lighter shade. And then you have, these are the ribosomes. The ribosomes are attached. The small and the large units. You have your messenger RNA, and these are the ribosomes, and these are the polypeptide chain, the protein chain that has been synthesized. That's how it happened. You have your, in this case, there's the DNA, okay, and then this will be the messenger RNA, and then this would be the proteins that is being synthesized. What you would notice is that in prokaryotes, because there are no membrane-bound organelles, the the sites of protein synthesis and the sites of messenger RNA synthesis are all in the same area. They are in the cytoplasm. So the process of transcription, that is the making of messenger RNA, and the process of translation, the making of protein, all take place at the same time. They take place simultaneously. Because when the messenger RNA is being synthesized, it is available for it to be translated immediately. So that the process goes together. In eukaryotic cells, however, because you have a membrane-bound organelles and you have a nucleus that has a membrane around it. So you have a nuclear envelope with pores through which molecules can be moved to the outer part, the cytoplasm. What happens is that you have the process of transcription taking place within the nucleus, and then the messenger RNA is then moved into the cytoplasm where translation or protein synthesis will take place. So there's a difference between the two. The process of transcription, the making of messenger RNA, would take place in the nucleus in eukaryotic cells. The process of translation, the making of proteins, would take place in the cytoplasm. For prokaryotic cells, the two take place simultaneously. When eukaryotic cells synthesize messenger RNA, it is not a final product. It's not a finished product. It has components called introns. So there's an intron, that's an intron, and the introns are non-coding regions. So it has to be prepared, it has to be processed before it is moved to the outside or into the cytoplasm. And the processing happens because there are enzymes which help with the process of splicing. So this intron will be excised, this will be cut off, and this will be cut off and removed. The same for this intron as well, cut off and removed. And then the rest are then bound together, glued together. Now, this is a more final product. When it's almost done, a 5' cap will be added to the 5' region, and then the 3' end, you have what we call the poly A tail will be added. These molecules, what they do is that they make the messenger RNA more stable and less liable to be degraded once it's being transported into the cytoplasm. So you can see some differences between the two in terms of transcription and translation. By way of review again, replication for prokaryotes would take place in the cytoplasm. For eukaryote would take place in the nucleus. Replication is making DNA copies, and it happens during cell division. Then transcription, the making of an RNA, for prokaryotes would take place in the cytoplasm. Remember, it has no membrane-bound nucleus. For transcription in eukaryotes, it's going to take place in the nucleus or the nucleolus. Nucleolus, it's a very dense area within the nucleus where synthesis of RNA take place, ribosome synthesis take place. Then the process of translation, which is the making of proteins from RNA, would take place in the cytoplasm for both prokaryotes and eukaryotes. You have, for DNA in prokaryotes, you have a region called the untranslated region. So this is the untranslated region. You would have sequences of nucleotides where the polymerase will recognize and be able to bind to. And that area is called the Shine-Diagonal sequence. So you have sequences there that makes it easy for the ribosome to bind to. This sequence will be transmitted to be processed and copied and transmitted to the messenger RNA. And you need this point on the messenger RNA, the Shine-Diagonal sequence, right, for the ribosomes to bind, both the smaller and the larger subunits to bind. Okay, so that's something I want to mention there. But yeah, Very similarly, you have the messenger RNA transcribed from DNA and then translated into a protein. So the strand of DNA from which the messenger RNA is made, the complementary nucleotides are made, is known as a template strand. So that's the template strand. The strand that it is not made from is the non-template strand. I want to add this other information. You will notice that the sequence of the base pairs here, the sequence of the nucleotides here, it's the same as the sequence over here. You just pay attention and look at it. You see G is G, U, you have T because you don't have you don't have T in RNA. G is G, U and T, A and A, C and C. So we call this strand, the non-template strand, is also called the sense strand. Okay, so this is the sense strand, and this is the nonsense strand. The nonsense strand. Okay. You have the promoter, so that's the promoter, and then you have RNA polymerase, so this is RNA polymerase. Notice the difference between DNA polymerase and RNA. DNA polymerase is needed for replication, RNA polymerase is needed for transcription. So the shine diagonal sequence that I was mentioning, this is it, and that's where the ribosome will attach to, and it's very close to the start codon. So shine diagonal sequence and the start codon are needed for the process of transcription, for the process of translation, the making of proteins from messenger RNA. So this brings us to the end of part 2 of our lecture on microbial genetics.